Thank you. That's one of the nicest introductions I ever had. I should have brought my apron. Once we finished synthesis, I got a nice apron, which was, what was it, Plastic Chef? Yeah. Yeah, so I should have brought that, but okay. It's a future perspective, but it is actually also looking back because we found that the creation of standards for micro and nanoplastic research is really an emerging topic, I would say, within the field. It's something that we thought we all understood already, and or like Juliette actually said, would be easy, right? I mean, you order some particles and you start your toxicological assays, for example, but that was not the case. And I think the underlying challenge here is that plastics are often considered as a single term, a plastic, whereas plastics are, of course, as you are likely aware, much more complex. There are different shapes, different sizes, different crystallinities, polymer types, and then there's also another group which we don't even really focus on too much, I think, within Aurora, but that's the additives. Think about plasticizers, stabilizers, scavengers, pigments that give them their color. So these are all very important to consider if you want to understand what every specific type of micro or nanoplastic brings as an hazard or might induce as a risk. And we found in the beginning that there was no diversity at all in these micro and nanoplastic materials. So this is a literature-based assay, and we were focusing on fluorescent particles because we wanted to look at uptake models, so the particles should be fluorescent. We found that 83% of all the literature was focusing on polystyrene. And this was because polystyrene was commercially available. It has been used and developed as a standard for calibration purposes in analytical chemistry and detection methods for years. And although we found an increasing number of publications on micro and nanoplastics, unfortunately, the ratio, which is the light blue line of polystyrene studies, remained stable. So even this year, I checked, and people are still first going towards the polystyrene commercial spheres because they are readily available. So within Aurora, we decided that we wanted to do this in a different way. Why? As I said, the complexity of plastics, just shown here in the plastic waste distribution of 2015, which is where the literature essay started, let's say, shows you that polyolefins are mostly present. But as Amanda also showed you, we found mostly polyesters, or for example, nylon polyamides. So polystyrene is actually not representative for the whole pool of materials that we have. And the shape, not even to mention that, spherical particles are likely the most unrealistic thing you will find in nature. So, chemistry, which is what I love. You see beakers here, which is what I have seen a lot in many different colors. But please don't go into details. Feel free to catch me later or during the Q&A, and I can give you all the details that you would like. But what is important is that this emulsion polymerization is what's making polystyrene particles. And it's very workable. So the polystyrene particles are made in perfect spheres, and it works like a charm, but only for polystyrene. And this explains why polystyrene is dominating the market. The emulsion polymerization technique doesn't work for other polymers, with a small exception perhaps for PMMA. But if you try other polymers, it becomes very complicated. So we decided to go for nanoprecipitation method. And this is what some people call bottom-up. I would always call it reshaping, because theoretically you're taking a large piece of plastic, dissolving it, and then casting it into nano or micro-sized particles, whatever you would like. There's a third one that's not on this slide, which is physical degradation. So there are also within CUSP partners that we're looking at, for example, cryomilling, even ball milling of plastics, making really degraded micro and nanoplastics to study their effects. But within Aurora, we decided we wanted to focus more on specific parameters to understand these. So therefore, we needed something that was reproducible and although more realistic, also in that sense, still within defined limits. So that we could say, for example, it's the size, or it's the polymer type, or it's the shape of the particle that has a certain effect. So the real deal. We synthesized 5 polymers. I would say the major ones, the polyolefins, PET, PMMA, PVC. And you can see them here in these nice electron microscopy images. The histograms show you a particle size distribution. These are the particles that we synthesized, and they are all around 100 nanometers, which is a vital size if you think about, for example, transportation over physiological barriers. Like I said, there was still a lot of parameter control. So the size was, the shape was definitely not spherical, but spherical-like. They were flaky or irregular, which would be more realistic. But in that sense, all the particles were still comparable. So that allowed us to make a proper comparison between the samples, as Juliette already showed. And we found these indications that polyamide might actually trigger real effects. And therefore, I was asked to synthesize polyamide. Polyamide often degrades into a fiber. So what we tried is actually making it as a particle. Why? Because then again, you can compare it to the other particles. Size, shape would not matter, and you're really looking at polymer effects. So this is what we tried in terms of creating standards. And as already been shown, these work quite nicely. So within the vivo cells, we see uptake of these particles. We added fluorescent markers. We tested these to be very stable, that they don't leach out, for example, and then you can actually trace them back within cells. It even went so far that we were adding markers which were the same to every polymer, but would react differently within a different polymer structure. So the markers would even be the same within the whole batch of testing materials, but they would give you another output signal, which means that you can identify actually mixed exposures without changing a marker. So you don't have to worry about, for example, that your marker is giving specific toxic effects. This is what we have learned. And then this slide is only 3 bullets towards micro and nanoplastic standards. I think what we have learned and what I honestly believe in, and what we think as AURORA would like to advocate for the whole micro nanoplastic field, is that reference materials or standards, they should be controllable. So it does not really work if you have a wide range of micro nanoplastic in your jar, which you did not characterize fully, because you might get effects, but you don't know where these effects actually come from. So scale, polymer size, polymer type, these are the things that you should know and you should control. And nanoprecipitation might be a very nice way to do this. Robust markers are essential. If you add fluorescence, it should not leach out, because the fluorescent dye might have an effect, something that you really want to avoid. So rigorous testing should be standard if you make these particles. And then finally, accessible methodology. What we mean with this is we want to create a lot of these particles, and it should be easy to do for many people, because not everyone in this interdisciplinary field has a strong chemistry background. Ideally, we create methodologies that everyone can make their own micro and nanoplastic particles, and therefore within different projects, also for the future, create a case study which is comparable through all these different projects. That would be fantastic, and that would really harmonize the field. So that would be our recommendation from AURORA, thinking about standards for the future. Thank you.